Okay, let's take a look at nephron today. What is nephron? Nephron is a basic functional unit of the kidney. The nephron is made up of Bowman's capsule, proximal convoluted tubule, loop of Henle, and also distal convoluted tubule. Okay? Collecting duct is not part of the nephron. Okay? Collecting duct is connected to multiple nephron. Here you can see one collecting duct over here. It is connected to one nephron, two nephron, three nephron, four nephron, all this. Okay, so collecting duct is not part of the nephron. So let's take a look at the simplistic view of the nephron first. Here you can see the nephron. The first part of the nephron is the Bowman's capsule. Okay, Bowman's capsule surrounds the glomerulus. So I will label this as glomerulus. Well, this is your afferent arteriole and this is your efferent arteriole. Okay, so if I can show you another diagram, which is over here, this is a more realistic view of the nephron. Clear all the drawing first. Okay, so here you can see we have the blood vessels, the afferent arteriole, blood coming in from the afferent arteriole, then go into the glomerulus before leaving through the efferent arteriole. If you are small enough, it's possible for you to pass from the glomerulus and into the Bowman's capsule. Okay, so I want to clear. Okay, so this is the Bowman's capsule. Then subsequently, this is the proximal convoluted tubule. Then after that, we have the loop of Henle. And then we have the distal convoluted tubule over here. And finally, this is the collecting duct. Okay. This is the realistic view. And here is the simplistic view. Okay. Bowman's capsule. Proximal convoluted tubule. We call it proximal because it is in proximity to the Bowman's capsule. That means it is much more nearer, so proximal. While this one is distal because it is in a distance away from the Bowman's capsule. They are both known as convoluted because they are highly coiled. This is also highly coiled. So proximal convoluted tubule, also known as PCT. Distal convoluted tubule, also known as DCT. Then we have the loop of Henle. This is like one loop, one and only loop. And finally, the collecting duct. Okay. Let's take a look at ultrafiltration. Before that, I need to let you know the glomerulus, which is the one in red here, the glomerulus, which is this one, and the Bowman's capsule, which is this one in orange here. They are collectively known as renal corpuscle or Malpighian corpuscle. Okay, so this structure here, renal corpuscle, its main function is to carry out ultrafiltration. So how to carry out ultrafiltration? So basically, if you look closer, we have drawing first. If you look closely, the afferent arteriole, which is this one, has a larger diameter. It is slightly larger than the efferent. Okay. So when the afferent arteriole's diameter is larger than the efferent arteriole's diameter, this will cause large amount of blood content to get stuck over here. Okay, you imagine two red blood cells can come in, but only one red blood cell can go out. So another red blood cell will be stuck here. And then two more red blood cells come in. And each second, only one red blood cell can go out. So more and more red blood cells get trapped here. Okay, creating a what we call hydrostatic pressure. So the hydrostatic pressure will then drive ultrafiltration. Okay, thanks to this ultrafiltration, blood is pushed through a three-layered membrane of the renal corpuscle that contain pores and slits that allow selective movement of molecules and ions. So they will selectively allow things that are small to pass through. Okay, things that are large will not be able to pass through. So this is not in your syllabus. Okay, normally they won't ask you what are the three layers of renal corpuscle, but this is just for your information. Okay, the one in the box. Okay, here, those that are able to pass through, those that are not able to pass through, these are tested in your syllabus. So you should take note. Those that can pass through: glucose, amino acid, water, mineral salts, and urea. Okay, the main thing that we want to pass through, we want them to pass through is urea. Okay, red blood cell, platelets, and plasma protein cannot pass through because they're too large. Okay, so how is ultrafiltration achieved in Bowman's capsule? Okay, this question is almost the same as the one earlier, but this is focusing on Bowman's capsule. Okay, while this one is Bowman capsule and glomerulus. So you can use the afferent arteriole and hydrostatic pressure, all this. But Bowman's capsule is just this, what we call orange structure over here. Okay, it doesn't involve the glomerulus, which looks like another puddle of capillary. So we can't really talk about the afferent arteriole and efferent arteriole. So if the question asks you about ultrafiltration in the Bowman's capsule, then you have to talk about how the capsule is lined with layer of cells that contain slits that allow small particles to pass through, but not large ones. Okay, simple as that. So ultimately, blood is forced through the membranes of the renal corpuscle. 1/5 of the blood will pass into the Bowman's capsule, forming the glomerular filtrate. So take note, those that manage to pass through the glomerulus into the Bowman's capsule are called glomerular filtrate. Okay, because the glomerulus is acting as a filter paper, so like glomerular filtrate. The rest of the blood, which is 4/5 of the blood, will exit the glomerulus through the efferent arteriole. Okay, next, I need to teach you on the difference of selective reabsorption and also secretion. So what is selective reabsorption and what is secretion? So you need to take note. Earlier, I mentioned that ultrafiltration has occurred. So if you are used to be at the blood over here, through ultrafiltration, you manage to pass through and you go into the Bowman's capsule. You are now part of the glomerular filtrate. So if you did not get reabsorbed, you will continue your way and move and move and I speed this thing up. And then eventually you become part of the collecting duct, and eventually you go to renal pelvis, ureter, urinary bladder, and then through the urethra and come out as a urine. Okay, this is provided you are not reabsorbed. Okay, so in other words, reabsorb refers to when you move from the renal tubule and into the blood. If you're moving from the blood and into the so-called, color is not good. How about you move from renal tubule into the blood? This is reabsorb. On the other hand, if you move from blood into the renal tubule, this is secretion. Okay, simple as that. Okay, we call this reabsorption because the same nutrients have been absorbed once from the intestine into the blood. So this is the second time it is being absorbed. So we call it reabsorb. Okay, finally, let's take a look at the main function of the region and also the molecules reabsorbed, the molecules secreted, and also what are the molecules left. Okay, so we start off with Bowman's capsule. Okay, this is not really correct. So, okay, things that manage to enter the Bowman's capsule would be glucose, amino acids, mineral salts, vitamin B, C, water. Okay, so they enter into the Bowman's capsule with the help of ultrafiltration. In the proximal convoluted tubule, selective reabsorption will occur. Selectively reabsorption will reabsorb all the glucose and amino acids, 70% of the sodium chloride, and 70% of the water. Okay. Proximal convoluted tubule will also actively secrete urea, uric acid, and creatine into the renal tubule. Okay. So at this point of the time, you expect, at this point of the time, we still have, basically we have removed all the glucose, no more glucose already. No more glucose already. Amino acid, no more amino acid already reabsorbed. So there are still some mineral salts left, some vitamin B, most of the vitamin B, water, and okay. So after that, let's take a look at the loop of Henle. Okay, in the loop of Henle, you have the descending loop. The descending loop, which looks like this, the descending going down, descending. The descending loop is permeable to water, so it will only reabsorb water. And then we have the ascending loop. The ascending loop is only permeable to salts, so only sodium chloride is reabsorbed. Water is not reabsorbed. Okay. Then finally, let's take a look at the distal convoluted tubule, also known as DCT. In the distal convoluted tubule, selective reabsorption of calcium ion, sodium ion, and also water under the influence of ADH, which is antidiuretic hormone, Antidiuretic hormone, we will explore this in the chapter known as hormones. Okay, we won't explore it here. So selective reabsorption of all these three will occur, while active secretion of urea, uric acid, creatine, hydrogen ions, potassium ions, ammonium ions will occur where these substances are secreted from the blood and into the renal tubule. Okay, so then what is left in the glomerular filtrate? Now we have mineral salts, vitamin B, C, water, urea, and also the ions that is secreted into the glomerular filtrate earlier. Finally, we have the collecting duct. Remember, the collecting duct is not part of the nephron. Collecting duct's function is there to collect filtrate from nearby nephron. Okay, so at this point of time, you can still reabsorb water back under the influence of ADH. And these are the substances left in the glomerular filtrate. These are the substances that enter the collecting duct. Okay, so this is the final review here. You can see what are the stuff that, let's start from ultrafiltration through the Malpighian corpuscle. So after the glomerular filtrate enter the proximal convoluted tubule, PCT, sodium chloride are reabsorbed, water are reabsorbed. H plus, K plus, NH4 plus equals ammonia, and also urea will be actively secreted into the tubule. That's why you see the direction is from the blood into the tubule. And then amino acid, glucose, and vitamins are reabsorbed back, but not all of the vitamins. If there are too much vitamins B, C, then it's not all of them are reabsorbed. And then finally here, the descending loop of Henle, only water is reabsorbed back. And then here, the ascending loop and ACl salts are reabsorbed back. Then NaCl is reabsorbed back. H plus, K plus, and NH3 is secreted. Ammonium is NH4 plus, ammonium ion. So it is actively secreted into the distal convoluted tubule. Water is also reabsorbed back. Then finally, at the collecting duct, you can reabsorb back water. Okay, very simple. Okay, lastly, let's take a look at frequently asked question. Okay, how is selectively reabsorption achieved in the proximal convoluted tubule? So here you can say that the cells are packed with mitochondria. With a lot of mitochondria, then you can release large amount of energy for active transport. Okay, another frequently asked question is about the length of the loop of Henle. So the animals that need to conserve water normally have a longer loop of Henle. Okay, why? Because if the loop of Henle is longer, then you can absorb more water. Absorb, 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 absorb. As compared to another loop of Henle, which is shorter. You can only absorb possibly half the amount of this. Okay? So you can follow this way of answering. Then lastly, how is urine formed? Okay, when you want to tackle this question, make sure that you have your key points. What are the key points? Number one, you must talk about ultrafiltration. Number two, you must talk about selective reabsorption. And number three, you must talk about active secretion. These are the three main points that the teachers are looking at. Okay, so that's it. That it is for today. I'll see you guys in the next video. Bye-bye.